So once we've got these mice cells inside the epithelium of the small intestine, then these are packaged into what we call chylomicrons. And it's through these chylomicrons that we can actually enter the lymphatic system, which is what we see here in the green. So the lacteal will link in with the lymphatic system, and from there we can actually deliver the triacylglycerols into the bloodstream. So there's a little bit more detail here about this process. So basically, within the epithelial cells, we have the reassembly of the triacylglycerols into what we call the chylomicrons. So the chylomicron is a combination of the triacylglycerol, phospholipids, cholesterol, and also proteins. Essentially, what the chylomicron is it's a carrier for the triacylglycerol. So remember, the triacylglycerols are nonpolar, so they need a carrier to be able to carry them through polar environments. And remember, the body is mostly made up of water. So the chylomicron is the lipoprotein that will carry the lipid through the lymphatic system onto the bloodstream. And the triacylglycerols are the major constituents of these chylomicrons. So we can't go directly into the capillary walls because the chylomicrons are actually too large. So instead we have to go through the lacteal, which is a vessel of the lymphatic system, and then onto the thoracic duct, and eventually into the subclavian veins to enter the bloodstream. And once we're inside the bloodstream, as the triacylglycerols, you can then be absorbed into cells and start using them from there. So once we do have the triacylglycerols in the bloodstream, we break those down into the fatty acids in the glycerol, and this is done by enzymes called lipoprotein lipases, so enzymes that specifically break down our chylomicron, and the fatty acids are then able to be absorbed by the body cells, and once they've been absorbed by the body cells, they can really be used for two things. Either they can be broken down into constituents to get energy, so the fatty acids will be broken into acetyl-CoA, and the glycerol will be used to create the dihydroxyacetone phosphate, which connects with our glycolysis process. And the other thing we can do, instead of utilizing the fatty acids in the glycerol to derive energy, we can actually store them in the adipocytes. So basically, these are the cells where we store the triacylglycerols, and it's just basically a very large droplet made up of many triacylglycerols, and this is what we have inside our adipocyte cells, which form the adipose tissue or fat tissue. Alright, so we can see a summary of this process here. So we start off with the dietary triacylglycerols. There is no effect of the saliva on the digestion because the saliva contains polar enzymes and the lipids themselves are nonpolar. Once we get into the stomach, then two things happen. The fat droplets will actually start to disperse into the chyme, and we'll also have partial breakdown of the triacylglycerols into fatty acids and monoacylglycerols, thanks to our gastric lipases, which begin to break the ester bonds which connect the triacylglycerols. So remember, there's an ester bond connecting the fatty acids and the glycerol. So we then take that content into the small intestine, and this is where the majority of the digestion takes place. We use the bile to solubilize those droplets, so it's going to be more stable in the polar polar environment of the small intestine, and we've also got other pancreatic lipases which further break down the triacylglycerols into monoacylglycerols and fatty acids, which are then able to assemble together to form those micelles, which is basically that ball with the polar heads around the outside and then the fatty acid tails pointing inward. So the micelles can actually be absorbed into the intestinal cells, where we then repackage the triacylglycerols into chylomicrons, and once in the chylomicrons via the lymphatic system, we can actually get those chylomicrons to enter the bloodstream, where they can then be broken down into triacylglycerols. The triacylglycerols can then be broken down or hydrolyzed into fatty acids and glycerol, and we then have the fatty acids and glycerol in the bloodstream, ready to be taken up by cells and then either used to create energy or possibly for storage. So there's a lot of different steps here, but the most important processes occur in the small intestine. This is where we're getting the majority of the breakdown and the repackaging, which allows us to go from the small intestine into the lymph and then into the blood. And then once we're in the blood, we can break down the triacylglycerols into the fatty acids and glycerol, which can then be used in different metabolic processes. And that's what we're going to be talking about in today's session, what we actually use the fatty acids for, and also what we use the glycerol for. So where does the majority of the digestion take place from our dietary fats and oils? So we do have digestion occurring in the stomach, but it's less extensive than in the small intestine. So in the small intestine, we have our pancreatic lipases breaking down our triacylglycerols to form the monoacylglycerols and also the fatty acids, which combine together to form micelles. So that's where the majority of the digestion is happening. So what has to happen to get the breakdown products in the small intestine into the blood? So we have to repackage the monoacylglycerols and fatty acids into micelles, which brings them into the intestinal cells, and then we can package those into what we call chylomicrons, which are able to move through the lymphatic system into the blood. So once we have our triacylglycerols in the blood, what can we use them for? Well, firstly, we'll break these down into the fatty acids and also the glycerol, and in this form they can be absorbed by cells and then either used in metabolic processes to derive energy in the form of ATP, or we can store these as triacylglycerols in the adipocyte cells. So here we can see storage of the triacylglycerol. So basically we've got a huge number of triacylglycerols within this yellow droplet, and you can see that that's taking up the large amount of this adipocyte cell itself. So most cells do have a limited ability to store triacylglycerols. So for this reason, we use these specialized cells called adipocytes, which are very good at storing the triacylglycerol. And the majority of the cytoplasm is actually made up of the triacylglycerols. So where do we have these adipocytes? It's beneath the skin and also around the vital organs. So depending on how much adipose tissue you actually have, then you may have quite a bit of excess fat in these locations. There's especially the fat around vital organs can actually be quite dangerous and have poor health outcomes. So one advantage of having the adipose tissue is though, you're actually insulating yourself, so it's actually easier to retain heat in cold environments and can also protect organs from physical shock if there is sudden impact. So just like in our carbohydrate metabolism, what we're going to do with our triacylglycerols is actually under the control of hormones. So our glucagon and also adrenaline can trigger the breakdown, the hydrolysis of our triacylglycerols. And they do this by activating an enzyme called hormone-sensitive lipase. So it's a lipid-breaking enzyme that is activated by hormones. So that's where it gets its name, because the enzyme is activated by a hormone. We call it hormone sensitive, and it's a lipase because it will break down lipid, specifically our triacylglycerol. So it splits the triacylglycerol into its individual components. So remember I showed you that triacylglycerol contains our glycerol and those three fatty acids. So essentially what the hormone-sensitive lipase is doing is it's cutting these ester bonds that connect the glycerol and fatty acids. So we're now going to have separate fatty acids floating around, same with our glycerol. So they've all been separated. So now that we've got this separation, we can actually use the fatty acids and glycerol for different metabolic processes. So the breakdown of our triacylglycerols, or also known as the triacylglycerol mobilization, is actually an ongoing process, and about 10% of your total triacylglycerols are actually replaced daily. So triacylglycerols represent the majority of our energy reserves, also known as fat reserves. So the majority of the energy that we store is actually in the form of triacylglycerol. So we can see that represented in this table on the right here. So almost 85% of our energy storage capacity is actually in our triacylglycerol. 15% in protein, about 0.5% in our glycogen. So remember, this was our carb store, so very low amount of carbohydrate we can store, and blood glucose is a tiny portion as well. So the majority of the energy storage capacity that we have is actually in the triacylglycerol. So where do we derive the triacylglycerol from? What type of energy source? So remember, this is our dietary fats and oils, which are made up of many, many triacylglycerols. So if we have too many triacylglycerols inside the cells or in the blood, then we're going to start storing these in our adipocyte cells, which will form the adipose tissue. Alright, so once we've got that triacylglycerol and we've undergone the hydrolysis to break it down into the two individual components, we're left with one glycerol, and we're also left with three fatty acids. So this slide tells us actually what is happening to the glycerol, because we actually have different reactions for the glycerol and for the fatty acids. So we'll actually have the glycerol being taken up by the liver or the kidneys, because these are the organs that have the capacity to convert glycerol into something that can be used to derive energy. So in these organs, we have the enzymes present that can convert our glycerol into our dihydroxyacetone phosphate. So this is a two-step reaction. Firstly, we actually have to phosphorylate our glycerol, so turning our glycerol into glycerol 3-phosphate, and this actually costs us an ATP. So we're taking the phosphate off ATP and adding it onto our glycerol. And the second step is an oxidation, where we're actually turning our alcohol group into a ketone group. So this is facilitated by the oxidized coenzyme, which is actually being reduced to the electron-rich version. So we don't go into this process in a huge amount of detail. Takeaway messages for this is that the glycerol is actually converted into the dihydroxyacetone phosphate, which allows it to be incorporated into these metabolic processes of glycolysis and gluconeogenesis. So these are both associated with carbohydrate metabolism. So essentially, the glycerol is entering carbohydrate metabolism by getting converted into the dihydroxyacetone phosphate. And remember, this occurs in the liver and the kidneys. So we're able to utilize our glycerol to actually turn it into something useful, which can be used to generate ATP if we're using glycolysis, or if we're doing gluconeogenesis, then it's going to be used to make glucose. Okay, so this is a connection between one type of metabolism, our lipid metabolism, and our carbohydrate metabolism. So we'll see as we go through these lipid metabolism lectures, there's quite a few connections between the two. So that's one of our components, one of our breakdown products of our triacylglycerol covered. So we saw that the glycerol is actually used to turn into the dihydroxyacetone phosphate, which we can use in glycolysis or gluconeogenesis. But what about the fatty acids? We actually need a different reaction to make use of these. So the fatty acids are actually broken down through a oxidation process, and the oxidation process that we do is called beta oxidation. So beta oxidation is a process which takes place in the mitochondria. So there's a few different steps to this process. So firstly, we actually have to prepare our fatty acid for our beta oxidation. So this is the activation of the fatty acid, and you can think of this as a preparation step. We then actually need to move the fatty acid into the mitochondrial matrix, because that's where the beta oxidation takes place. So we actually have to transport it to the correct location. And the last part is to actually do the beta oxidation of the fatty acid. So for our beta oxidation, we actually have the input being the fatty acid, specifically the activated fatty acid, and the output is many acetyl-CoA. So why is acetyl-CoA useful? What can we use it for? So we can use the acetyl-CoA to link into the citric acid cycle, because it's needed for the first step there. And through our products of our citric acid cycle, which are electron-rich coenzymes, we link with our electron transport chain and complementary process of oxidative phosphorylation, which eventually will allow us to make a lot of ATP. Okay, so this is the primary use of the acetyl-CoA that we get from beta oxidation. We can use it to eventually make lots of ATP. So the activation of the fatty acid actually takes place on the outer mitochondrial membrane. So remember the structure of our mitochondria. We've got the outer membrane, and then we have the ruffled inner membrane, and the ruffles form what we call the cristae, which is basically the folded part of the membrane, and then in the center we have the matrix. So the space between the outer and inner membranes also has a name. Do you remember what this name is? So we call this the intermembrane space. So it's definitely worth making sure you understand the structure of the mitochondria, because a lot of the processes that we talk about in metabolism actually refer to the location in the mitochondria. So it could be the matrix, it could be the outer membrane, it could be the inner membrane, or it could be this intermembrane space. So here we see that the fatty acid activation actually takes place on the outer mitochondrial membrane. So here is the mitochondrial membrane. So it's happening on this outer mitochondrial membrane. So essentially what has to happen for the fatty acid to be activated is it has to attach to our CoA. So remember, we've seen CoA before in the form of acetyl-CoA. So the acetyl group, which we see here in blue, attaches to the CoA, and that allows the acetyl-CoA to enter the citric acid cycle. We actually want to attach a CoA to the fatty acid, which we see represented in blue here as the carbonyl group connected to a long group of carbons and hydrogens, which we have on our fatty acid, represented as R here. So we want to attach our fatty acid to the CoA because this is what we need to do to activate our fatty acid and actually start the process of beta oxidation. We need to do this. So to do this actually requires both of the phosphate bonds within ATP to be broken down. So the ATP will actually get converted to AMP because we cleave these two phosphoanhydride bonds, and that will release lots of energy. And this energy is allowing us to overcome the energy barrier needed to attach the CoA to the fatty acid. So essentially, we need to do our fatty acid activation so we can create what we call the acyl-CoA, which is the combination of the fatty acid and also the CoA. And this is the first thing we have to do before we can start our beta oxidation.